Right, here's where we're at. We chatted a little bit about water, pH, hydrogen bonds. Next, what I wanted to discuss with you is the very basic chemistry of the molecules of life and some other things. So molecules of life, what do I mean by that? Pretty much all life forms, well, not pretty much all life forms, consist of polymers, right? Polymers are the molecules that are composed of smaller building blocks. Like when we talk about polyethylene, it means many ethylene molecules linked together. And that linkage changes the properties, properties of those molecules. And there are several major types of, for goodness sakes. Just give me 1 second. Carbohydrates are going to be the first molecule, life polymer that we're going to talk about. Carbohydrates are chemical compounds that consist of carbon, hydrogen, and oxygen. This is the empiric formula. You don't have to know the empiric formula. What you have to understand, though, is that there are so-called monosaccharides and di- and polysaccharides. When you have several monosaccharides, like glucose, galactose, or fructose, you can link them together in the reaction of dehydration. You essentially remove water. And this reaction of dehydration produces the bond between two monosaccharide residues. You stick them together like Lego pieces, and then you have disaccharide. By adding new and more pieces of monosaccharide, you can create polysaccharide molecules. Does that make sense? Okay, so in the reactions of dehydration allow you to stick together monosaccharide residues, creating large polymers, either linear or branched. In plants, those large polymers are starch. You know them as starch. In animals, it's glycogen. Cellulose is also a carbohydrate, the polysaccharide. Does that make sense to you? Okay. So dehydration allows us to make polysaccharides. The thing is, they are so big that in the form of polymer, they are pretty much useless. We have to break them down back again into the individual pieces, into monosaccharides, to do something with them in the cell. Now, if we build them, if we link monosaccharides in the reaction of dehydration, to break them down and take away water, to break them down, we have to add water. And that water addition can be achieved by the reaction of hydrolysis. Does that make sense? Hydrolysis. Okay, let me see if I can find any program here that I can use to picture things. Probably the easiest will be to run the paintbrush. Yeah, paint. Okay, just I can write down. Really, I can write down. So here's what hydrolysis would mean. So I have one molecule, another building block. So I have polysaccharides, right? It's a polysaccharide. And then you add water, okay? And this bonds are gone. Does that make sense? By adding water, you hydrolyze them. You release monosaccharides. With monosaccharides, you can actually do something, okay? Now, what is the major function of carbohydrates? By far, energy. Yes, it's the main energy source. Is that the best energy source? In terms of energy density, like per gram of sugar, can you think of anything else that gives you more energy than 1 gram of sugar? There's actually something. Fat. Fat is more energy dense. But biochemically, sugars are easier to work with. Let's put it this way. They can directly get into the main metabolic pathway, the glycolysis, and be used for energy production. Fats, they have to jump through multiple hoops. Okay. Fats are great to store energy, actually. Okay. Bacteria use some other compounds to store energy. We'll talk about it later. Now it's not really important. What you have to now, step by step, understand is that carbohydrates are the energy supplying molecule. Doesn't mean that they don't have any other function. They're also parts of the structural elements like glycoproteins or glycolipids. Okay, and of course ribose and deoxyribose, two pentose sugars, are parts of RNA and DNA molecule respectively. So you can say they are indispensable for genetic information storage. Next molecule that we're going to talk about is lipids. It's not molecule, it's a class of molecules. What is, if you will take fats, like, which fats do you know? Which fats do you have in the kitchen? Oil, like cooking oil, something else. Shortening, margarine, butter, yes. So you see they can be liquid or solid. So all these things that you just mentioned, oils, margarine, butter, they are all triglycerides. We call them triglycerides because they have one molecule of the alcohol named glycerol, which has 3 hydroxyl groups. These 3 groups can form bonds, chemical bonds, ether bonds, to 3 molecules of fatty acids. Okay, so you have 3 fatty acids and 1 glycerol molecule as building blocks for the triglyceride molecule. Okay, so this is your triglyceride. Now look at the reaction here that's illustrated here. Again, you have dehydration. You take away water. Okay, here's water that you take away. You get triglyceride. What do you need to break triglyceride back into glycerol and free fatty acids? Add water. How do we call this reaction? Hydrolysis. Does that make sense? You will see that when we polymerize something, when we build something up, we take water away. When we break it down, we break it by adding water. Make sense? Triglycerides serve as pretty much energy source. Yes. No, those are, the great question, those are covalent bonds. So it's a chemical bond, which is pretty hard to break, to be honest. So if you want to hydrolyze fat in the lab, you have to place it in most likely very basic, very alkaline environment. And I would bet you have to boil it. And then you can separate it into the glycerol and free fatty acids. Enzymes in your intestine can do it without all of it. They much better than we are. So, but that's the covalent bond between carbon and oxygen. Absolutely. So the major function of triglycerides all over in the world, in the nature, is the energy storage. Another kind of lipid, phospholipid, consists of the glycerol, two molecules of fatty acids, and one phosphate residue. Now, if you would look at the phosphate residue here, it has negative charge, for example. Phosphate and glycerol, when they kind of combined, they form what we know as hydrophilic head of the molecule. And fatty acid tails form hydrophobic tail. So you have a molecule that has a hydrophilic, okay, and hydrophobic parts. And that makes them the main component of what? Cellular membrane, right? Because your membrane becomes hydrophilic outside and inside, where it is in contact with either watery environment or watery cytoplasm. But it's hydrophobic where the tails are. You see tails here? So it's like a hydrophobic layer in disguise. Okay? You also can break it down, of course. Phospholipids, you can break it down to phosphate, glycerol, and fatty acids. Okay? Now, cholesterol and hapine, or hopanoids. So sterols and hopanoids are molecules that regulate the strength, the rigidity of the cellular membrane. Sterols can be found in eukaryotic cells, in the membranes of eukaryotic cells. Hopanoids can be found in the bacterial membranes. Okay? Now, any idea why cells, bacterial cells, or protozoan cells, let's focus on microbes, may need to regulate the rigidity of the membrane? To adapt, what kind of pressure would you think? Not literally pressure, but what kind of environment changes could it be? I mean, what would cause, when changing the rigidity of the membrane would be needed? Like, why do you want to change it? Why don't just let it stay at the same level all the time? If it becomes, say, softer, will it become more penetrable? So you can alter transport, right? You can alter transport across the membrane. If this is a protozoa, eukaryotic organism that does not have cell walls, so it has a soft envelope, it may help to adapt to changing pressures, okay, or maybe squeezing through the blood vessels or something like that. So that there are certain biochemical signaling may be changed by changing the membrane rigidity. You kind of lock membranes in a certain part of the membrane. Don't let them go away. So transport and signaling can be affected by those molecules. And pretty much the difference, along with the structural, is that sterols are in eukaryotes, hopanoids are in prokaryotes. Bacteria, not prokaryotes, bacteria. The third macromolecule is the protein. Any questions on the previous one? Not yet, I suppose. Proteins. If you have okay. Have you taken biology like 1200, 1510 before? Have you? How many amino acids you were taught about? Anyone remembers the number? That's like classical. Huh? 20. 20 amino acids in the proteins. That's classical way. Turns out there's 21. Okay, 21st amino acid was discovered about 25 years ago, early 90s. Yeah, yes, about 25 years ago. Not really good with math. So the 21st amino acid, just FYI, is called selenocysteine, and it's like cysteine here, but instead of sulfur, it has selenium. Different atom. It's actually a growing area of biochemistry now. They study why it's so important to have selenocysteine. Anyway, so if 21 different amino acids doesn't mean that every protein will have all 21 amino acids. Okay, but you have an option. You have options, right? Now, amino acids linked together, again in the reaction of dehydration. You see, water is taken away, and amino group and hydroxyl here form the peptide bond. By sticking different amino acids one to another, you can form a chain of amino acids. Does that make sense to you? So again, it's really, I think, it's like a train. If you see, the train usually does not consist of only one type of a car, right? Different, tanks and other things that I do not know names for because I'm not a trainologist. So different types of cars, right? And they have certain order, which makes sense. So what happens for protein is the same. You have different amino acids that form that primary sequence. Now, important thing about amino acids, look at them. These 6 amino acids shown here, they all have something in common. This carboxyl group, amino group, that alpha carbon here, that's all the same. Look, no difference, right? No difference. But then you look, there's a side chain, and side chains are completely different between 21 amino acids. So the side chains, the chemical structure is what defines, in the end, the structure and the function of protein. Does that make sense to you? I'm a sucker for analogies, but I don't have a good one here. Seriously, I just don't have one. So when you put those amino acids together, you form so-called primary structure. Now this chain of amino acids, it doesn't stay straight. It starts to fold. Okay? Pretty much spontaneously fold by itself. The only analogy that I can suggest is the coil. You can straighten it up, right? But when you release, it's going to go back to the coil form because it's low energy form. For protein, folding is the way to achieve, to get to the low energy form. Is that clear? So there are 2 major secondary structures, alpha helix and beta sheet. And when alpha helices and beta sheet fold together, they form tertiary structure. Proteins are very, very complex molecules, structure-wise. Let me put it this way. We have enormous computational power. Do you agree with me or not? My first computer, which my parents bought me in 1996 when I was first in college, didn't have money to do that myself, was like 1,000 times weaker than this thing. What should I say? First computer that we moved to States 10 years ago, Dell laptop. was worse than this phone. So now we have enormous computational power. You've heard about supercomputers, right? We can model things and do amazing stuff. So let me put it this way. If you have a protein which consists of, say, 100 amino acids, there's not enough computational power in the world to calculate its shape. We have all the equations, we have all the physics, we have all the methods. It's not enough computational power in the world combined to calculate protein shape in the reasonable amount of time. So by the end, the calculations will be done, the humanity will be over. That's how complex the interactions are. That's pretty amazing. What we can do, we can predict that this part of the protein is most likely alpha helix. This part, another part is beta sheet. That's about it. We can't really fold it. That's pretty amazing. You know what people do now? Have you heard about the project that searches for extraterrestrial life? When you can download the software in your computer, it's like a gigantic network. They analyze massive amounts of astronomical data. There is now a project called Fold It. You can download software, download the data of the protein sequence, and you start to fold. Just playing, just folding it. And system can analyze which shape of the protein is more preferential. So thousands and thousands of people play this game, folding actual structures, and they already solved several structures of the proteins. They already said, okay, this is how it should look like, this is how it should look like. It's pretty amazing. People have nothing to do with biology. They just play for kicks. Now, for many proteins, tertiary structure is the last one. That's like a globule, okay? Some proteins get into the quaternary structure, several globules put together. You definitely know one protein of humans that have several parts. It has globin in the name. Hemoglobin, yes, it has 4 separate protein globules interacting with each other. Not linked, but interacting. Okay. Here's one very important feature of proteins, the feature of denaturation. If you cook eggs for breakfast, well, at least some of you, I suppose. Do you? When you break it and you put it in a skillet, how does it look like? Egg. Just okay. Like a snot, right? Kind of sort of. Actually, in Russian, we call it a snotty eggs. Okay. Now, when you continue cooking it, when I was in high school, I never heard this expression before my high school, and my classmate asked me, "Do you want eggs like a snot?" I said, honestly, no, but I'm curious what it is. It turns out, like over easy, kind of sort of over easy, because they look like a little bit of a snot there. Anyway, when you cook it, they become more and more solid. Okay, less and less transparent. Okay, something definitely changes. Not only water goes out of the eggs, but also proteins denature in that egg. Okay, if you put, that's kind of blood, if you get blood on the piece of clothing, I'm not asking where did you get this blood, okay? And you want to wash it away, which water should you use, cold or hot? Cold. God forbid. Hot water will denature proteins, they will stick to the clothing, and it will be a problem to come off. Does that make sense? It's all about denaturing. Denaturation is essentially changing the shape of the protein. But one example that I love, girls will understand it. Perm. Perm. When you get perm, what they do with your hair? Chemicals and heat. Chemicals denature proteins, heat denatures proteins. So they pretty much torture them and change their shape. What happens after a while with perm? They go back. Proteins assume their normal shape in the hair. So hairdresser denatures them, they go back to normal. Same is applied when you want to straighten your hair. Normal shape is curly, you apply chemicals, they will straighten up. Does that make sense? Why I talk so much about denaturation? Chemicals like acid, base can cause denaturation of the proteins. Same goes for temperature. If you denature proteins, can they perform the function? There you go. Become dysfunctional. So that's really important to understand. Environmental alterations will affect the function. What are the functions of the proteins anyway? What do we need them for? Not primary. We can use them for energy, sure. It's not the main big functions. Yeah. We are protein life forms. Protein-based life forms. Everything on Earth is protein-based life forms. Structural, right? Something else. Magic E word. Starts with letter E. Mention that word today. The reactions. They run fast. Because of enzymes. Yes, enzymes. Vast majority of enzymes are proteins. Okay. So that's two main big functions of the proteins. Any questions on proteins? Nucleic acids. Well, you have 4 types of nucleotides, A, G, T, and C. I would love to picture. I will try to fix it during the break. A, G, T, and C in the DNA, A, G, U, and C in the RNA. Don't focus too much in that complementary. Well, they complementary, right? Anyone remembers pairing? It's kind of there. A pairs with T, G pairs with C. This sounds like a trivial thing, but it's really a fundamental and very important law of life. Because it means that each strand of DNA, and we have 2 in the molecule, has all the information about the genome. Here's the provocative question. Technically speaking, completely hypothetically, would 1 strand be enough? Yeah, why not? A lot of viruses have only 1 DNA strand, single strand DNA genome. Again, hypothetically speaking, can you use RNA to encode genetic information? Sure. Technically, nothing prevents you from that. The only reason why longer living organisms use DNA is much less prone to mutations. RNA is very unstable molecule. Okay. So pretty much the only function of DNA is to store genetic information. I tried to think of anything else, and I could not. Really. Just couldn't. Okay. Now, few important features. What's going on? The DNA is that it can be copied. So that genetic information can be transferred to the next generation of cells. Okay? This is fundamental thing. A little small digression. Remember we talked about microbiology breakthroughs on Tuesday, like how we used to have just, I'm lazy and I also proponent of the careful use of antibiotics, which is a synonym of being lazy to go to the quick care and get antibiotics for my sore throat. We used to it. You have sore throat, you go, you get antibiotics, you're fine, right? 20th century, not even 100 years. Anesthesia, the first I checked, about 1840. First anesthesia. Before that, 2 bottles of vodka, then you partial anesthetized. Okay, so we take a lot of things as they were known forever. Okay, the nature of heredity was not known till like late 1800s, early 1900s. Not nature, just idea of characteristics being, inherited by other organisms and stuff like that. DNA was much later. The idea of DNA storing that information, it's pretty amazing. Mendel, when he was establishing his laws of heredity, had no freaking clue about the genes or DNA. Absolutely not. Just didn't know. So it's profound property. Now, this information that's in DNA not only can be stored and transmitted, it also can be expressed. For this we use messenger RNA. RNA is single-stranded molecule, but it can fold. What I mean is, when it's synthesized as a single strand, it can form a freaky structure. Well, not like this, but it will start to kind of, one part of the strand will interact with another part. They will form like hairpin loops and other interesting structures. But RNA is single strand. Okay, and then RNA becomes a template in the process of protein synthesis. Which non-membrane bound organelle synthesizes protein? Anyone? Non-membrane bound organelle, really small, that makes proteins in the cell. Ribosomes, yes. So ribosomes bind RNA, produce proteins. Now, proteins confer traits. They are the reason why we look like we look. Okay. Okay, here's the good thing. So we just figured out that DNA is the carrier of genetic information, right? So genome, what is genome? What is human genome? Is mouse, huh? Not almost characteristics. It's information about characteristics. Okay, so genome has information about all the traits that we have. If I would take Mackenzie's genome and Courtney's genome, sorry, I don't remember all the names, so I will have to address it. I'm sorry. If I would take Alex's genome and Courtney's genome, Mackenzie's genome, it doesn't really matter, you're both blondes, okay? You look quite different. If we would compare the genomes, would they be profoundly different? There will be very, very few differences. It's going to be all the same. Pretty much all the same. Now, I don't remember if we talked about the expression. Yeah, we talked about expression, I think. If we would take genome of me and genome of chimpanzee, Well, I hope we look quite different, like profoundly. Sometimes, though, I may expect we're very much alike. The difference between us on the genetic level would be 1%. Not a lot. So why genomes of two people who look very different are identical? What defines how we look? What defines what we have? Hair color, eye color, I don't know, everything. What defines? Turns out it's not just a set of genes, it's how they are expressed. So gene expression defines the phenotype, the set of characteristics. Does that make sense? So I may say that we all have different phenotypes. I will be right. We have different height, different weight, different, I don't know, skin color, hair color, eye color, okay? I don't know, maybe, like I'm short, I have nearsightedness, okay? I have flat feet, somebody else may have some other things, I don't know, pancreatitis, for God's sakes, God forbid, no. Or, I don't know, gastric ulcer or asthma, okay? It's all about expression of those genes. Does that make sense? Now, here's one of my, so this is the expression. This is called central dogma of molecular biology. DNA is transcribed into RNA. RNA is translated into proteins. Proteins confer traits. You are blue-eyed or gray-eyed? Blue-eyed? Blue-eyed. I'm brown-eyed. It means I have more melanin produced from the same melanin genes that Mackenzie has. Okay, that's it. During our lifetime, that may change. Child may be born with the gray eyes because melanin production is low. As child grows up, eyes become brown because melanin production increases. You see what I'm trying to say? So, not all genes are expressed constantly. Okay? Actually, the expression, the phenotype, will be defined by the conditions in the environment. We can observe it in the lab using microbes fairly easily. One of the most astounding implications of that fact, that environmental pressure changes the phenotype, is development of antibiotic resistance. If you put a bunch of microbes, and think about this, microbes are very, very small, okay, and they replicate fast. Speaking of fast, what is the generation time for humans? When we say this was 3 generations ago, okay, how much time is that? 1 generation. 25 years. Because look, every 25 years, people give birth, like in around age of 50, you become grandparent. Okay, well, it varies, but on average, it's about 25 years. What's the generation time for bacteria? What do you think? No, too fast. Too fast. Way too fast. Huh? Hour is kind of average in the organism. On the plate, we talk about tens of minutes. The fastest in the lab is Campylobacter jejuni, 13 minutes. Every 13 minutes, the population doubles. If you will start counting, it freaks you out very soon, because if you imply that you have a bacteria with 15-minute generation time, and you start with 1 cell, in 10 hours, it's going to be 2 in the power of 40, which is about 1 trillion cells, which is the actual weight. It's like about 1 pound of bacteria. If they have resources, they would do it. So bacteria replicate very fast, which means their DNA is copied constantly. Does that make sense? When you copy something on, frequent basis, what will accumulate in the copies? Like rewrite with your hand. Huh? Mutations, right? Mistakes. It will accumulate mistakes. Those mutations, they can be good, they can be bad. Okay, now this is something that I want you to understand about evolution. When we say bacteria evolved to do something, no, they didn't. They didn't evolve to do. Evolution does not have a direction. What happened? There were many types of bacteria. Some of them survived. Some of them didn't. Right? So there are different phenotypes. Some of them survived under the selective pressure. Let's call it having good mutations. Some of them didn't. Those that had bad mutations. So the genetic changes allowed them to adapt to the pressure, like antibiotics. So you have a population of bacteria, millions and millions of cells, and some of them have mutations that confer resistance. You see that orange one? And then you put that bacteria on the medium that has antibiotic. Antibiotic will kill the bacteria that are not resistant, but resistant ones will survive. Does that make sense? And then resistant ones, of course, will take over, and you have a population of entirely resistant bacteria. That makes sense? So you can do it in the lab. You can start with a low dose of antibiotic, plate bacteria. Some of them will die, some of them will not. You collect living ones, continue plating, increasing the dose of antibiotics. Okay? So you build up the resistance. Have you heard about, medieval times? People, like kings and queens, they got poisoned a lot. One of the ways to avoid being poisoned is to do what? Huh? Say again? Yeah, that's the way. It's a good way, but then food tester would die. How can you avoid that? Like skip the food tester. Yep. You start with a small amounts of, not with every poison you can do that. But with some toxins, you absolutely can do that. So you can start with a small dose and increase it. And people manage to develop quite a good tolerance to poisons. The only problem is that the killer uses different one, then you're screwed. Pretty much it's the same way bacteria use. They get used to the pressure. Does that make sense? This is an example of evolution, and here's the famous saying. It's almost annoying now. Biologist Theodosius Dobzhansky once said, "Nothing in biology makes sense except in the light of evolution." And when we will talk about things in microbial metabolism, microbial genetics, microbial growth, we have to keep in mind evolution. We have to just think, why does it happen this way? They evolved. What was the selective pressure that routed them towards this? That makes sense? Always keep in mind the evolution. Now, here's my favorite analogy that I love it. I'm so proud of myself. When you think about DNA, RNA, and protein, those concepts are pretty vague. You didn't, well, except for the egg, but you never actually like touched it, never saw the actual process, right? I didn't, like physically. So what I compare it to is, imagine you have a cookbook. Recipe book. Not just recipe book, the recipe book. It was in your family for 2 centuries. The recipes from your grandparents. Okay? I don't know, written by, I don't know, print on the printer, on the paper, typewriter, all kinds of paper, all kinds of recipes. Only beef stew, for this you have like 37 of them. 15 different apple pies. Huh? That's a lot, right? That's a lot different apple pies. So you have like, I don't know, thousands of recipes there. If you need to make a dinner, really nice dinner for a party, are you gonna cook everything that you have in that book? Probably not. So what you're gonna do, you're gonna select a couple of entrees, salads, I don't know, some appetizers, desserts, and then cook this, right? Here's the problem. The book, twice the size of that notebook and like triple the thickness. It's huge. Holding it around in the kitchen is a pain in the butt. Another problem, it's your family heritage. It's the treasure. You don't want to bring it into the kitchen, risking to spoil it, to, spill something on it, right? So, here's the task for you right now. Really, think about the book. What would you do? There you go. Well, I would take my phone. Just snap a picture. I can cast the picture on the TV screen and the TV in the kitchen. Just look at the picture and then do all the cooking. Right? That's easy. So this book is your DNA. A copy of the recipes that you want to cook is your RNA, and the dinner is the protein. Does that make sense? Protein that you express right now. Menu right now. Okay? Maybe for Sunday brunch it's going to be completely different. Right? So think about this. Think about expression. Now, we start talking about microscopy today. Microscopy may be a little bit of staining. I don't know how far we get. I want to talk about microscopy. Maybe we'll leave the staining for later. So, my microscope is better than yours. Sorry. Seriously speaking, conceptually it's not very much different. Slightly different objectives. Mine's connected to the computer, so I can project the image there. And it has one more capacity, the so-called phase contrast, that yours doesn't have. Other than that, they're pretty similar. Okay? So microscope is based on the refraction and reflection of light. Refraction through the transparent material or reflection from the mirror. Okay? So refraction happens when light crosses a lens. Right? So you may have convex, concave lens. They either make the focal point farther like concave or closer like convex. So you magnifying glass, will have convex lens. Okay? And the purpose, of course, is to make things that we cannot see, naked eye, visible. When you were kids, when I was a kid, I used the magnifying glass to look at something really, really small. Okay? Probably you did too. Now, microscope kind of brings it to the next level. The microscopes that you're going to work with, and this one too, is this microscope is light. It weighs a lot actually. It's not that light, but light is the source of radiation here. It uses light. It's compound, which means it has several objective lenses. It's binocular, 2 eyepieces, and it's parfocal. Yours are the same. Parfocal means that when you switch objectives, your image remains in relative focus. Relative, not perfect, but relative. Make sense? Every microscope, or should I say every time you work with a microscope, you should think about 3 things. Magnification. Magnification is how bigger the image is. doesn't mean that it's going to be clear, right? So magnification, say these lenses, the eyepiece here, it's 10x magnification. Yes, 10x. Okay, which means that they increase the size of the image as it appears 10 times. That makes sense. Objective lenses that you're going to have are 4x, 10x, 40x, and 100x. Total magnification of the microscope is calculated as eyepiece magnification multiplied by objective, right? So the maximum magnification that you can get there, you have 100x, so 1000. 10x on your eyepiece, 100 on the objective, 10 by 100 is going to give you 1000. We'll get to that. It's oil immersion. Yes, it's oil immersion, yes. So we will use it, but not all the time. Okay. That was magnification. So I understand how to calculate it, right? Does that make sense? If you're confused, tell me. Because I cannot, thank God I cannot read your minds. Resolution is how clear the image is. Resolution is the ability to distinguish between two closely located objects. Resolution is mostly defined by the wavelength or frequency of light. Okay? Here. So these waves have low frequency and large wavelength. These waves have high frequency and short waves, right? So the higher the frequency, the better the resolution. Which light has the highest frequency? Which part of the visible spectrum is the highest frequency? Huh? Opposite. Blue. Yes. Red is the longest in the visible spectrum. Okay. So we try to produce the light that is closer to the blue end of the spectrum. Does that make sense? If we would be able to see in UV, that would be even better. Okay? Now, resolution, again, magnification and resolution are not directly linked. One of the best examples of it, if you watch any crime scene investigation show, that kind of show, you know, when they look at the footage from security cameras and there's a license plate, and it's like really crappy photo of the license plate on the security cameras, and they blow it up, and then you can like read everything, even see minute scratches. It's bullshit. It's not going to happen. Try get any picture on your computer screen and blow it up. It will become grainy, more and more grainy, right? So resolution is limited for every microscope. Now contrast is the difference, the brightness difference between the object that you observe in the background. What objects are we going to observe under the microscope? Bacteria, cells, essentially. And what are cells made of mostly? What is the main chemical component of any cell? Water. And our cells that we're going to look at, what are they going to be floating in? Water. So you have to separate water from water. Well, think with a little less water from water. Believe me, contrast is not going to be amazing. Images just by themselves, those images pretty lousy. I mean, there's not a lot of contrast because those cells, the organelles in those cells, if there are any, will absorb very little light. So yes, they will be darker than the background, but not very much darker. Okay? We will talk how we can enhance that contrast. So these are first microscopes. That's the Leeuwenhoek microscope, which is kind of funny. And this is the microscope suggested the kind of makeup suggested by Robert Hooke. Remember the guy? It looks very much like the modern ones conceptually. So Hooke looked at the quark and he actually saw the cells. He never connected cells and life. Okay? Hooke was a scientist. Leeuwenhoek was a respected Dutch glassmaster. So when he developed the first microscope, what do you think he looked at first? Eyes, huh? Say again? No. No. Huh? For this he developed telescope. You're too romantic. No. That's kind of bland. It was exciting but not romantic at all. Even well, that's very romantic. Wine is romantic. Huh? I don't think there was any tequila at the time. It was like 1600s. His own poop. He was really curious what's in there. Huh? His own poop. His own feces. And he actually found some protozoa there. He pictured them. So he had some intestinal infections. It was exciting, yeah. Now, here's the parts of the microscope. So, from this one, you have to understand these names: compound, binocular, parfocal, and blah blah blah. Difference between magnification, resolution, and contrast. How we calculate magnification, okay? Now, parts of the microscope. I will ask you about what are the main function. Well, eyepiece, look in them, huh? Say again? Yeah, to see through them, exactly, with just our eyes close to them. Objective, lenses, focus on the specimen, okay? Objectives are mounted on the revolving nosepiece. Stage, this thing. Stage can be moved by the knobs here, okay? They're called XY mechanical stage knobs, okay? The good thing about it is that you don't have to use your hands. Your hands are much less precise than those knobs. Right? Make sense? Light comes from the light source. It's called illuminator here. Fancy word illuminator stands for the lamp. There's a little lamp. Light that comes from the lamp goes all directions. So condenser, which you can't really see and you don't really have to worry about. Condenser focuses the light on the specimen, produces that if you will see the spot of light, right? And diaphragm, you will have a little running thing. You see this thing here? That's the diaphragm. You can open it and close it, regulating amount of light, and actually when you decrease amount of light, you increase contrast. Okay. Well, this wheel, you will have wheel. I have on this side wheel rheostat, regulates how bright the lamp is. And here are your focusing knobs, probably the most important thing. So I have bigger one, which is coarse focus, and the smaller, which is the fine focus. So you start with a scanning objective, which is 4x, okay? And you find whatever you want to look at. And you use coarse focus to bring it to focus. Then you change to the next objective, okay? Which is going to be 10x. So total magnification will be 100. Now you use only fine focus knobs, but not coarse. The reason is, and your image is going to be in kind of sort of focus. So you use fine focus knobs to adjust it just a little bit, okay? And then you can switch to higher magnification. And again use only fine focus. Why do you not use coarse focus knobs on high magnification. Well, slide is okay, they're dispensable. What's worse? Lens, yes, you can crush the lens. Make sense? Also, magnification is so high, you turn coarse focus and you completely lose the focal point. Just completely, you absolutely out in the blue. You have to go back. Does that make sense? So types of microscopy. We're going to go over the types of microscopy till staining, so till this, okay? And then we'll take a break and we'll look at what we've got without hand washing. So, bright field microscopy. We're going to use only bright field in this course. That's the simple one, not that it's not functional or inferior. You have specimen on the bright background. In this image, you see bacillus microbes. They are additionally stained, so they're bright red. But even without staining, you will be able to see the microbes. Okay? Cool? Terrific. So you can see that on the bright background. The most common. Does that make sense? Just simple to go method. Dark field is useful for observing live specimen. In this case, there's some trickery with the diaphragm. Pretty much blocks the light almost entirely. So somehow light illuminates cells, but not the background. You see, background is kind of dark. Okay, so in this case, you don't have to stain microbes. You increase contrast so you can see them floating around. Because when you stain microbes, they die, essentially. So this is Borrelia burgdorferi. I don't know if you can see, they kind of spiral. Okay, that's a spirochete that causes Lyme disease. Phase contrast. FYI, when I use word fancy, it usually means either very expensive or something that I personally don't know how to work, and it's how it works and it's magic for me, or both. So phase contrast is fancy, not very expensive, but fancy, light trickery that by using interference allows you to increase contrast even without staining. I will prepare a slide and put it in my microscope, and those of you who want, you can go and look what phase contrast microscopy looks like. It's slightly better than bright field, but not that much better. Okay, so you can see, you see specimens and sometimes internal structures. See, that's without staining, which is pretty amazing. Differential interference contrast, DIC, allows you to obtain very high resolution image with high contrast, and image appears almost in 3D. Now again, here you have some light trickery that, don't ask me, no clue. Fluorescence. Did you ever have those t-shirts that glow in the dark? Okay, that's fluorescence. You use special stains that you add to the microbes, to the cells. And when those stained cells are illuminated with the UV light, they start to glow. It can be useful, for instance, in identifying very small microbes. For example, mycoplasma, microbe that causes infections of the respiratory tract and your genital tract, is very, very small. It's really hard to see in a regular light microscope. Company Herbst developed special staining that stains exclusively mycoplasma. So what you can do, you can stain your sample for mycoplasma, then light it up with UV light, and cells of the mycoplasma will appear as glowing spots. You would never notice them using regular bright field microscopy, but you will at least see those dots, okay, that fluoresce. You can also stick the fluorescent molecule to antibody that will recognize a specific microbe, and you can pretty much diagnose the disease by using immunofluorescence. Now, if you never saw the fluorescent proteins, have you heard about fluorescent animals? Animals that glow in the dark, like green rabbits or green mice. They now sell fish. You can buy a fish, it will glow in the dark. Have one? Okay. That's the, huh? Scientists did it because it, out of the science, well, first, glow in the dark green rabbit is cool, right? It's just cool. Second, the idea to show that you can actually modify an animal so all cells will glow in the dark, or you can get it delivered only to the skin and hair. So it's more of a tool exercise, like developing tools. It's very useful. And look, I bet some people would pay probably $1,000 for glow in the dark rabbit. Huh? Yeah. So, oh, not at all. Now, believe me, those rabbits, the lab rabbits, they would not survive a minute out in the wild without glowing in the dark. So, yes. I see what you're saying. Not really, no. In MRIs and body scans, they inject special contrast agent that shows up in that specific, should I say, radiation, okay? You can, it's not the same. The purpose is similar. Does that make sense? The purpose is similar, but that's not the physically the same, chemically the same things. Okay, so you can use antibodies to differentiate different viruses, bacteria, protozoa, and stuff like that. So this is actually some bacteria stained with the dye. Confocal. Bringing MRI in a conversation. When they do MRI, what they essentially get in the end, the doctors? Like the whole body MRI. How does it look? What they can reconstruct? Yeah, pretty much the whole body. It's like 3D image. They can go slice by slice, right? That's what they do, slice by slice. Confocal microscopy uses pretty much same principle, okay? Changes the focal points and takes slices, quote unquote, okay? Various images at various depth, essentially kind of slices of the cell, and they can be put together by the software and produce three-dimensional image. It's hard to believe that this is three-dimensional. That's kind of a lousy picture, but it can produce sort of three-dimensional image of the cell. Usually you have to stay in this. Two-photon microscopy. Now this is fancy in both terms. We had a two-photon microscopy system in the place where I worked to study blood cells binding to the walls of blood vessels in the mouse. So it was in the live animal with live cells stained with fluorescent dye. Here you see the example of the intestinal cells stained with fluorescent dyes. So essentially it's kind of a combination of fluorescent microscopy and some other trickery. Okay, so these lasers allow you to distinguish different cells in the live specimen, especially when the specimen is deep, because penetrating somewhere in a living tissue is kind of a complicated thing with light. That microscope that we used, well, I never got to use it. I just saw other people doing that. $250,000. Yeah. 250,000. I was shocked. Never knew it's money. Now that was light microscopy. This is electron microscopy. In the light microscopy, we use the beam of light to go through the specimen, right? Make sense? In the electron microscopy, we use the beam of electrons. That's the whole difference. Electrons allow you to increase resolution, right? So you can see smaller details of the specimen. There are two major types that I want you to know: transmission, electron microscopy, and scanning. Transmission is going through the specimen, okay? Crossing it, transmit. So essentially what you get is kind of a slice. Like imagine you have a stick of salami, you cut it across, you look at it. Can you tell how long it is? No. But you see what's inside. Does that make sense? So that's transmission microscopy. It can show you, for example, what's inside the cell. This is the Ebola virus. Okay? But you can't say if it's shaped like this or like this. Just have a slice. Scanning microscopy produces wonderful images in which you can see the shape and the surface of the cell, like this Campylobacter jejuni cells. Okay, you have no idea what's inside of them, but they look gorgeous. So I usually say that transmission microscopy is ugly and useful. Scanning microscopy is gorgeous and sort of pointless. But if you would Google especially scanning electron microscopy algae, you will get stunning photos. It's unbelievable that this beauty can exist. It's just wonderful, really. So what you have to know about microscopy, it looks like a lot of info. Basic differences. Okay. Bright field, bright background. Dark field, dark background, live specimen. Phase contrast. Live specimen with very high contrast. DIC, live specimens, almost 3D image. Confocal 3D image. Fluorescent, well, kind of self-explanatory. Cells fluoresce. Okay. 2-photon deep slices, deep layers, thick samples. And transmission and scanning electron microscopy. What's the difference? Transmission looks inside, scanning scans outside. Two types that we don't talk about. I'm not going to ask about them. It's tunnel scanning microscopy, which gives you even better resolution, and atomic force. Atomic force always stunned me. You can move atoms. The engineers in IBM research branch somewhere in 1988, they used, I think, neon gas atoms to make the word IBM using atoms. There were like 40 atoms in the whole world. So they made IBM, just putting atoms in place. It's just mind-blowing. Okay. Now we're going to take a break, and then when we'll come back, we'll analyze what we've got with our